Welcome. Today, we're diving into something right at the cutting edge of science, a field called quantum biology, and it could completely change how we see life itself. We're talking about taking ordinary glowing proteins and turning them into tiny, sophisticated sensors that can actually work inside a living cell.
Okay, so here's our game plan. To really get a handle on this, we're going to build it from the ground up. We'll start with something you might already know about, glowing proteins, and then, step by step, we'll unpack the wild quantum mechanics that let us turn them into something brand new.
So first things first, let's set the stage. We need to talk about the amazing technology that made all of this possible in the first place.
For a long time now, scientists have had this amazing trick up their sleeves: fluorescent proteins. You've probably heard of the most famous one, green fluorescent protein, or GFP. Its discovery was so huge it won a Nobel Prize back in 2008 because it gave us a way to literally light up the machinery of life and watch it work in real time.
Now, how does this glowing thing actually work? Well, the basic idea is pretty simple. It's a three-step dance. First, you shine light of one color on the protein. That light gives an electron a jolt of energy, kicking it up to a higher level. Then, when that electron naturally relaxes and falls back down, it spits that energy back out as light of a different color. Boom, fluorescence. But it turns out there's a little secret, a hidden quantum event tucked away in that process.
And this, right here, is where things get really, really interesting. Scientists found out there's this weird quantum behavior happening that they realized they could actually control. So, under just the right conditions, that excited electron doesn't just fall back down. Sometimes it actually jumps over to a nearby molecule. When that happens, you get something called a radical pair. Now, all you need to know is this: you now have two molecules, and each one has a lonely, unpaired electron. And for just a split second, these two electrons act like tiny spinning magnets that become quantum mechanically connected.
So here is the absolute key. This quantum connection is incredibly delicate. I mean, super fragile. The way their little magnets are aligned, whether they're spinning in the same direction or opposite directions, can be nudged by even the tiniest magnetic fields around them. And here's the kicker. That alignment, that spin state, actually changes the final outcome. It slightly changes how much light the protein gives off in the end. And that insight led to a brilliant question. If a tiny magnetic field can change the light, could we work backwards? Could we watch the light? And if it flickers, use that to measure those invisible magnetic forces happening inside a cell? And that brings us to the big breakthrough. A couple of recent studies have shown that we don't have to just hope this quantum effect happens. We can actually build proteins to do it on purpose.
This is the moment where we leap from pure science to engineering. Instead of just using a protein that nature gave us, researchers are literally rewriting its DNA blueprint. They're making tiny, specific changes to make that quantum state more stable, to make it last longer, and to make its response to magnetic fields way more predictable and easier to measure.
And what's really cool is how we know this is a solid idea. Two different teams proved it could work, using two totally different methods. One group at the University of Chicago basically took an existing protein and learned how to control it with lasers. Meanwhile, a team at Oxford built a brand new protein from the ground up called Maglev. When you have two independent successes like that, you know you're onto something real.
So you might be wondering, okay, that's clever, but why does it matter? Why is making a sensor out of a protein such a huge deal? Well, it all comes down to what we're using now.
You see, most of our best quantum sensors today are made of things like diamond. They're amazing, but they're outsiders. They're foreign objects we have to physically poke into a cell, which can cause all sorts of problems. Protein sensors, though, they're insiders. The cell builds them from its own materials, using our DNA instructions with basically zero disruption.
This table really lays out the trade-offs. Okay, for right now, the diamond sensors are definitely more sensitive. But look at where the protein sensors win. They are perfectly biocompatible. And this one is huge, targeting ability. Because it's encoded in DNA, you can tell the cell to attach your sensor to any other protein you want to study. You can put your sensor exactly where the action is.
This quote really nails the paradigm shift. You have to get this. We are moving past just observing cells. We are now giving cells a new set of instructions, a blueprint to build their own tiny, advanced diagnostic tools for us.
A tool like this doesn't just let us do the old things better, it opens up a whole new way of asking questions about how life works at the most fundamental level.
Just think about the possibilities. We could actually watch a single enzyme do its job by sensing its magnetic field. Or we could see precisely how a new drug changes its target protein inside a living cell.
This is the kind of incredible detail we could soon have access to. Now, of course, the technology is brand new, it's not perfect yet. A living cell is a warm, chaotic, messy place, and that's a really tough environment for a delicate quantum state to survive in. So right now, the big challenge is to make the sensors more sensitive and to find clever ways to protect them from all that background noise.
But you know, we have every reason to be optimistic. History gives us the perfect example. The original GFP also started out with big problems. It was dim, it faded really fast, but decades of work and clever engineering turned it into one of the most important tools in all of biology. We are almost certainly at the very beginning of that exact same journey with these new quantum sensors.
And this leaves us with one final, kind of mind-bending thought. What we're seeing here is a shift from passively watching biology to actively programming it right down at the quantum level. This is so much more than just a new tool. It's a new frontier where we're starting to merge the rules of information with the actual machinery of life.